So today we will finish discussing and reviewing viral evolution and then discuss crickets. So first we will continue discussing the paper Topology of Viral Evolution in preparation of Rabadon's talk that will occur Monday, December 9th at the IMA. So recall that we can calculate distances between a number of organisms by comparing either a protein, DNA, or RNA sequences of the organisms, possibly using a modified Hamming distance where we just count the number of differences between the organisms. So you can compare these two and just count the number of differences between the sequence alignment, but weighting, so different mutations would likely carry a different weight. But somehow we come up with a distance between these organisms using some kind of weight with the sequence alignment, and then can now create a tree where those that are closest distance together are closest on the tree, those that are further distance are further away from that tree. But there can be a problem if we have a reticulation event where instead of just vertical transfer, so just having a mutation happening over time, we might have a mutation happening where two things combine within here. The two sources that we discussed last time included one, homologous recombination, where we might have a blue organism and a red organism, and these two might, so if we talk about viruses, maybe these two organisms affect and infect an individual, and that gives a chance for homologous recombination to occur, where you can therefore get a recombinant product that has both part blue and part red. The other method was via reassortment, where when the two viruses again infect the same cell, when these viruses then want to escape the cell, they can then go into create a new virus where now the red and blue intermix together, and so we would again have this reticulation event where this virus was created from taking a segment of the red and then other segments of the blue. So reassortment is another method for this reticulation events to occur. Any questions so far? We then took a look at influenza, and if we just take a look at a single segment, well, you can't have reassortment for a single segment, but one can look to see, do you have homologous recombination? Well, for the influenza virus, there was no higher homology for K greater than 0, and so thus there was none of this reticulation events where two things came together. So that meant no homologous recombination. But if we take a look at multiple segments, so still the influenza, if we take a look at multiple segments, then we do get the non-trivial homology for both K equals 1 and 2, and thus there is this horizontal transfer. There is a reticulation events, but via reassortment, since we saw there was not homologous recombination among the individual segments. We also noticed last time that in terms of this reassortment, that you don't just get an arbitrary reassortment. So we saw that these segments got grouped together, meaning that if PB1 and PB2, if we have our two viruses and we create then that third virus via this reticulation event, via reassortment, if PB1 came from one virus, well, then PB2 is likely to come from that same virus. They segregate together. versus maybe the NA may have come from this other virus here, so maybe we would have gotten the NA from a second virus, so they may not have segregated together. Arbitrary reassortments, most likely just many of them just do not survive, but you do get some that segregate together and some that do not, and so one can create a variety of different new viruses depending upon the reassortment. With this reassortment, one can also get higher dimensional homology, and so this is an example of a K equals to 2, something equivalent to the 2-sphere, where one can now look at this sort of complex reassortment among several different viruses in this avian influenza. So we did not get a chance to take a look at the HIV virus. The HIV virus is interesting in that it is a single segment. So we just have one segment as opposed to the 8 segments that we had for the influenza virus. So since we have only one segment, there's no reassortment, so we can't have reassortment if we only have one segment. But there is non-trivial HK. So for H1 and H2, there is this non-trivial homology, and so thus there is horizontal transfer, there are reticulation events, and those must come from homologous recombination since we don't have the reassortment. And again, we can see a non-trivial generator of H2 that tells us how several of these viruses are related in this complex pattern. Any questions so far? There are 2 measurements that one can calculate, and one of them is the topological obstruction, so TOP for short, and that's just the maximum barcode length in any non-zero dimension. So if the maximum barcode length is about 440 in dimension 1 or in dimension 2, that would be the top for the avian influenza. For the swine influenza, the length of the barcode is shorter, but by seeing that there is this persistent homology in either the first or the second dimension, it does tell us that there is no additive distance, so we don't have a nice phylogenetic tree where we just have mutations occurring over time. We also have a reticulation event creating non-trivial homology that persists long enough that it's not just noise. And it does depend, you can see, based upon the type of virus. So for dengue fever or for rabies, one doesn't generally have these reticulation events. And so then a nice standard tree will work for these type of viruses. But for many of the other viruses, the HIV, the avian influenza, then we do have these reticulation events via either reassortment for the influenza or via the homologous recombination for the HIV. So the topological obstruction, it's called topological obstruction because if your top 0 is not 0, sorry, that's a typo, if it's not 0. So if you do have a barcode that is different than 0, if it is extremely small, well, then that might just be noise. But if it is different from 0 in some persistent way, then there is no additive distance tree, then you do have this reticulation event. This top measurement is stable. So that means if I add a bit of noise to the data, then the length of the maximum barcode will not change. So if we had a very long barcode, we'll still have a very long barcode. If we had a shorter barcode, it'll still be short, but it still might have some persistence. If it looks just basically like noise or 0, then, So this thing is quite stable, and so it is a good invariant to determine whether or not we've got a reticulation event. The second kind of measurement that we're interested in is ICR, the irreducible cycle rate. That's just the average number of one-dimensional irreducible cycles per unit time. So we can sort of integrate over time the number of cycles, persistent cycles that we're having. And if we do this and average it per unit time, well, then this measurement, the irreducible cycle rate, is proportional to and provides a lower bound for the recombination reassortment rate. Biologists are very interested in the mutation rate of viruses. How quickly a virus mutates will tell you a lot about, its behavior and how quickly it might spread. So the mutation rate is quite important. The irreducible cycle rate gives you a lower bound to the type of mutations for recombination and reassortment. So it's not all the mutations, but the mutations that come from recombination and reassortment. This mathematical measurement can give us a nice lower bound and is often proportional to it as well. So this ICR is also an important measurement. So we can see with HIV there is quite a bit of homologous recombination that does go on, and so that tells us that this virus is mutating at a decent rate, if you want to call that decent. So the mutation rate in terms of just recombination and in terms of reassortment, this is a good measurement for that. Any questions there? It is also good to have a conversion between mathematics and biology. So we'd like to know what the mathematics means, and we'd like to also interpret the biology in terms of mathematics in order to formalize it and to be able to come up with good models and good simulations. So this table that came from that viral evolution paper basically relates the mathematical terms to the biology terms. So we've got the genetic distance, calculating some type of distance between these sequences or doing it over a time scale. That would be corresponding to our filtration value of epsilon, increasing our radius balls. Our beta naught, that was our number of clusters. So mathematically, that was our number of clusters. Well, in viral evolution, you're interested in your number of clusters. So at your scale epsilon, so depending upon what evolutionary scale. So in terms of these trees, there actually is an evolutionary scale for trees. You might be talking about the trees of entire organisms, or the trees over a very rapid time period for just a particular sequence. So depending upon how fine of a scale that you want to go to, that can be measurement of your epsilon, which also relates to the same epsilon over here. The generators of your homology will just be a representative of the cluster. We also talked about hierarchical relationships among our H0 generators, and that comes with our barcodes. We have two bars right here, and then one of them gets killed by merging with an edge to the other one, and so we can cluster them, do a hierarchical clustering, saying that. So these sorts of barcodes where one persists and one dies, by noticing which edge joins, which edge kills this and joins it to which one over here, we get this hierarchical clustering among our H0 generators. And that's exactly the trees that we're getting out over here, except for we may not, we may have more than just trees in terms of our H0, in terms of our H1. But for H0, for just among H0, we can still create these trees using this hierarchical clustering. Any questions there? Our beta 1 gives us the number of our reticulation events. So you have a reticulation event right here, and that creates an H1. So that would be our beta 1. And we can again think about the generators. The generators would then correspond to reticulation events. Our generators of H2, we took a look at a couple of examples of that, and that shows us examples of complex horizontal genomic exchange, either via reassortment or homologous recombination. And if we do have HK is not 0, for some persistent bar, so for some persistent bar, then we don't have our phylogenetic phylogenetic tree. And so that was our topological obstruction to having a phylogenetic tree, is if we have a persistent cycle for some k greater than 0. And our irreducible cycle rate was the lower bound on our reticulation events, so the lower bound on the number of homologous recombinations, or how fast the mutations occur via reassortment or recombination. So that basically finishes up our viral evolution. Any questions about viral evolution? Let us now discuss crickets in preparation of Gideon's talk on Thursday, December 12th at the IMA. What we're interested now, though, is nerve endings in the terminal ganglion of the cricket. So a ganglion is just a dense nerve center. The terminal ganglion is just one that occurs near the dense nerve center near the end of the cricket. Afferent terminals are, so you've got these hairs, they're connected to these nerves, which then go into the ganglion, the terminal ganglion over here, and so these are just nerve endings right here, these afferent terminals. And afferent just means that you have these nerve endings are transmitting impulses from the periphery into the central nervous system. So we have now a picture of our cricket, and we've got the hair connected to the nerve endings, which then go into the terminal ganglion. So this is basically our data. Is we know this hair. The biologists have determined that the hair then connects to these nerve endings, which then sort of branch out in terms of these tree-like structures where you've got a whole bunch of points out here that then can transmit signals up through into the central nervous system. So what we're interested in is the shape of these things and how they reside within the terminal ganglion. So here is a picture of a whole bunch of, so we've got now a whole bunch of these tree-like structures now creating this terminal ganglion, this very dense nerve region. You'll notice that it is color-coded. The color coding is that basically each hair is responsible for transmitting particular signals. So not the hair, but the hair then attaches to the nerve ending, and the nerve ending then transmits a particular signal, and the signal that it gives is actually air current direction. So it tells you the direction of the air current. And so depending upon where you are within this ganglion, if you're here, you might be transmitting this direction. If you're over here, you might be transmitting this direction. And so each different hair will be responsible for a different direction of air current. And then the way the cricket is able to determine the direction is that direction gets transmitted via the nerve ending to a particular location, and then that location is associated with that particular direction. Any questions here? So again, we're interested in our data is a whole bunch of these things here, and we want to understand the structure of this terminal ganglion. And that structure is related to the direction of the air current. So our data set is broken up into proximal data, so that's the closest 15% to the terminal ganglion, versus the distal, which is the furthest 30% from your terminal ganglion. For your proximal data set, this is broken up into long, medium, and short, and you can see we have a whole bunch of data points, 42,000 for the long, more than 27,000 for the medium, and 29,000 for the short data points. And so we've got long hair, medium hair, and short hair. The length of the hair is basically related to a number of factors, including its age, and that does have a relationship as to where it does embed inside, especially long versus medium and short. With respect to the distal data, we only have 6,000, and they all correspond to long. Getting this data is more difficult. You have to trace this thing, so we've got our long hair that then attaches to this nerve ending. The tracing is, much further. Notice it's even sparser, because here we're doing 30%, covering twice the distance instead of the 15%. And so it's a very sparse data set compared to the much more data set that we have for the proximal data. But that's the data set that we're looking at now. The proximal data set was filtered to first off remove outliers. There is always noise, there's always experimental error, so removing outliers is a good thing. But this data was also obtained using many different crickets. Whenever you're comparing data from different sources, you're going to get even more noise, so removing outliers, removing your least dense regions is a good thing to do. So that's the first thing, is to filter your data to remove your outliers that are likely related to noise or complications from getting your data from a variety of different sources, a variety of different crickets. The other thing is to remove basically redundant points in your densest regions. So now in your most dense regions you want to remove points just because it improves the computational speed. Having lots and lots of data points gives us, if we keep all of them, it's a lot to work with, increases the memory, it slows down the speed. So if we remove our densest regions, not remove the densest regions, remove some of the points in the densest regions, then the speed will be improved. If you remember the density measure where we take the K, the density is the kth closest points. So that's basically what they use in order to determine density. And so they removed the least densest point using this density measure, maybe doing the distance being 2 apart, or 3 apart, or 4 apart. as well as removing some of the points in the densest regions. But the important point to remember is that data is usually nearly always filtered to remove both outliers and also to improve computational speed. Another thing that's frequently done is to do computational simulations. And so there were comparisons done using a Gaussian mixture model. The main thing I want to point out is that computer simulations are frequently done in most of these papers that we've discussed. Simulated data to see if things, your experimental data is all well and good, but if we can also simulate it, we can see if maybe there's some issues that one also should have seen. This is particularly important because since we do have some very sparse data in the distal end, comparing that to the not so sparse data in the proximal end, one can then fill in some of these things with a Gaussian mixture model and see if maybe the sparseness is causing issues or not. The other thing that is, the main thing that is different in this paper is how it's computed, and that's using cubical homology. Cubical homology is actually often used. I'm actually surprised we haven't encountered it before now, because it's much better for memory requirements. When you have a lot of data points, cubical homology does not require nearly as much memory as the RIPS complexes that we've been using. Cubical homology is very similar. It's just instead of using spheres, balls, we now use cubes. So if we were working in 2 dimensions, we would use squares. Now we're working with 3-dimensional data, so our data points are points in R3, and so we use 3-dimensional cubes. And then, just as with the RIPS complex, with growing the epsilon balls, one can then grow, take small cubes, larger cubes, just grow the size of the cubes, so each side of the cube we just grow. And so the idea is very much the same in terms of we can calculate RIPS complexes as well using cubes, but we're now growing cubes instead of our balls. One of the reasons is that we can do bitmaps. We can put these cubes so that they lie on a nice grid, and then we can use that grid to now reduce the size of our data. Instead of working with each individual ball or each individual cube, we can work with these bitmaps instead, and the fact that we're living on a grid. To calculate persistent homology, they used this software here, which is available on Rozak's website. There is an entire book. It's actually a very big book. I forget the number of pages, 500, 700 pages, a very big book on computational homology. And they stick basically to the cubicle because it is computationally very efficient, especially in terms of saving on memory. In order to calculate the persistent homology, this software was used, the Cube Persistent MD, to find the cycles. So once the persistent homology was found, so you now know the number of cycles, they used Hom cubes in some software called Chomp, which can be downloaded from Rutgers, or it's also embedded within Sage. Sage is a very large software package similar to R and MATLAB that has many different features, and so people can contribute to that. And so there's a number of features within Sage too, one of them being the Chomp software package. Any questions on that? So cubical homology is something one should consider, especially if you've got memory issues. And it's also good for speed too. So now let's go ahead and discuss the results. In terms of our results, what we have is the H1, so we're looking at the cycles, the one-dimensional cycles here for the proximal, so we're looking at just the proximal data set now, for the long versus the medium versus the short data set. So it's been broken up into long, medium, and short. For the long, 4 persistent cycles were found. Now you'll note that there is some arbitrariness in terms of the choice of what is persistent. You know, this bar right here, so this black bar, was determined to be too short to be considered long, a long barcode. So if we take a look at the long data and take a look at what persists, we've got one that persists for 23 cycles, one for 19. So as we're growing the sides of our cube, so after 23 steps, this one starts at this point and lasts for 23 steps. We also have one that lasts for 19, so starts around 3 and then lasts for around 19. One that lasts for 13, one that lasts for 1, and then there's also the shorter ones, including about 15 that lasted only for 2, not even drawn here, 11 that lasted for 3. So there were a number of them, and it was chosen. There is some arbitrariness to this, that anything that was 7 or less was not persistent. Anything 11 or more is considered persistent, and you can see that there is this gap, and part of this gap is because outliers and stuff were removed. It was the chosen, so this is using the reduced set, removing the outliers and removing redundant things in the densest regions. So over here, this is considered noise in this region for both the medium, short, and long, and in this region is considered the sparse. It's not the sparse, the persistent. So these are the persistent ones. Everything in red is what's considered persistent. So for long, there are 4 persistent cycles. For medium and short, there are 3 persistent cycles. And the question now is, if we start now combining some of these data sets, if we combine some of these data sets, for example, are these 3 cycles the same as these 3 cycles? Well, let's combine and find out. If we combine just the medium and the short, so we're combining just the medium and the short in order to now create this new one, we can see that only 2 cycles persist. And so before we had 3 cycles for the medium of lengths 12, 13, and 14, as well as 3 cycles for the short of length 11, 13, and 14. Now by combining them, we've now increased their persistence. They're now of cycles 15 and 16. So they persist for longer, and now we've got only 2 of them. And it was determined that one of the cycles for the medium gets filled in with the short data, as well as one of the cycles for the short gets filled in with the medium data. And so 2 of these actually did come from here. It wasn't that we were creating new ones. They could have done zigzag persistent to see how these 2 data sets embed within each other. It's possible that all 3 of them may have been disjoint, but no, there aren't. 2 of them are disjoint and 1 of them are not. And one can see that by actually taking a look at the data. So if we take a look at the data, this is the data. The A is for just the small. The B is just the medium, so the medium is shown in blue, the small in red, and if you combine the 2, we've got medium plus small. Now, one is supposed to be able to see 3 cycles within the small group and 3 cycles within the medium group, and only 2 cycles in the combined medium plus small data set. And you can see the orange cycle pretty clearly. Here's an orange cycle, here's an orange cycle. You can see part of the purple cycle, part of the purple cycle, part of the purple cycle, and that's what's common in each of the 3 data sets, small, medium, and long. You can also see part of a gray cycle. So this gray cycle, which gets filled in by the medium data set. There's a gray cycle over here as well. If you want to see the gray cycles, what you'll need to do is download the Mathematica files. So the Mathematica files for all of these data sets are available, so you can download and rotate them. Here we'll just see one rotated view of the medium plus small. So we've got our medium plus small, and then if we rotate it, we can kind of see this purple cycle a little bit better. The orange cycle, which was very clear within this thing, is now not so clear within this perspective, but the purple cycle is much clearer here in the medium plus small. So if you want to actually visualize the data, I highly recommend downloading that software, downloading the data, the Mathematica files, and visualizing it within Mathematica. So any questions up through here? So that is the combining the medium and the small. And there is a question as to whether or not, in terms of how this stuff fills in, looking at how these embed within each other, can say one has to define what is a medium length hair, what is a small length hair. And when you look at how these two combine, well, maybe there really isn't a clear divide between medium and short, and maybe they should just be grouped as one object instead. So that can give you some information as to, is your grouping, is your choice of deciding what is a short length, medium length, or long length hair really making sense with respect to your data. We can now take a look at combining all 3. And so here, remember, A was long, B is medium, and C is short up top here. And now down here, we compare all the proximal. So this is still the proximal. We're not doing any of that distal yet, of the long, medium, and short data. We can now see that while we had 4 cycles here, and 3 cycles here, and 3 cycles here, well, we know that when we combined the medium and the short, we are only left with 2 cycles. When we now add in the long, maybe these 4 cycles were independent of these 2, but no, they're not. One of these goes away, 3 of these go away, and we're left with just one persistent cycle of length 15. So, and again, there's a bit of a gap between what's considered noise. Now, anything of length up to about 9 is considered noise, and they're just taking this one hole here. Any questions about that? We can now throw in all the data, including the distal data. Actually, sorry, we're not throwing in all the data yet. Now we're just taking a look at just the long, so ignore the medium and the short for now. If we take just the long for both the proximal and the distal, if we add in the distal, 2 of these holes get filled up, and we're left with only 2. That's actually kind of interesting, because remember, for the long, we had many points. We start off with 42,000. Then after reducing the data, I think maybe about 20,000 might have been left, after removing the noise as well as any redundant points in the densest regions. had still lots of points for the proximal data. For the distal data, we only had 6,000, so they actually did not do the persistent homology on the distal. They figured the data was just too sparse for it to really be valid. But if we now throw it in for both the distal and the long, 2 of these holes get filled up. And so in terms of our persistent, only 2 things are left to persist. We can then throw in all the data, and if we throw in all the data, you'll see that there's no red. So now there is nothing that really persists, and so basically everything gets filled in. We can try to, again, look at the picture, but it's a little bit hard to see in terms of the picture unless you can actually rotate it in 3-dimensional space. But here is long, medium, plus short for proximal. So on the left is all the proximal. And remember, in the proximal data, we had exactly 1 cycle left once we combine. And you can see that cycle pretty clearly. That's actually, it's kind of nice to sort of see this cycle. You can kind of see sort of a solid torus in here, and a solid torus might be nice because with this color coding in terms of representing, you know, if we do see this sort of torus, maybe that relates to, remember we're doing directions in terms of which way the wind blows. Is it blowing at this angle, or is it blowing at this angle? And so it's sort of nice to see a cycle within here. How well it's related, I think that needs to be looked at a lot more closely. But once one adds in the distal, that hole gets kind of filled up with these distal points. Oh, sorry, in terms of the color codings, to not get confused, the color coding for the long, medium, and short is green, blue, and red here. And then we add in the black for the distal. So for the distal, the black is now added in. And if you calculate the cubical homology, basically no cycles persist right here. You start filling in the hole that was for the proximal data set. I don't have the color coding for this perspective for the actual angle, so this is not the color coding for the angles. Exactly what the shapes mean still needs to be investigated, but once we now have the shapes, one can start thinking about what they mean a bit more. There is one additional interesting piece about this distal data. The distal data they have determined is that there is a time delay. So if you remember, in terms of we have to transmit the data from the distal hair all the way up into this terminal ganglion here. So that distal data has to be transferred all the way up here. And so there's like a 5 microsecond delay compared to data that comes from a hair in the proximal data. And so maybe in terms of the time delay, filling up this hole, that location might tell you more where in terms of that time delay comes from. So this is all a little bit hypothetical, but now that one has this complex, one can then try to understand in terms of where the long hair goes in, and now break it up in terms of the angle, which we have not done yet, to try to understand more in terms of the location and what the shape of this ganglion cyst here is. So ganglion, the terminal ganglion. It's not a ganglion cyst, that's something completely different, but in terms of the terminal ganglion, in terms of trying to study the shape of this. So any questions about studying the location of nerve endings in the terminal ganglion of crickets? Okay. Well, we will meet again on Wednesday for a live lecture. So the lecture will be live on Wednesday, but I will be giving it from a conference in Banff. So you can either listen to it via the Standard Iowa site, or there is a conference now going on the entanglement of biopolymers, and those talks will also be video streamed, including my lectures on Wednesday and Friday. So see you Wednesday and Friday from Canada.